it falls short, if it falls short of your expecting it, know this is on purpose. When we go into actually, most of you might be more interested in actually analyzing the models and trying to do, so that's not our goal today. Our goal is kind of to prime you into how you can build a draft model, something that you can get quickly, and it's hopefully is helpful to you, which is the other primary goal. We want to make sure that the models are helpful to you, and so that it's your starting point for your journey of curating and doing your own metabolic modeling analysis. So this that's the primer for today. So I'm just going to do a very quick introduction here. I'm going to have to glance at these subjects because I actually rather want to jump into the narrative and walk through there. But it's important that you just find some concepts here first for folks that might not really be familiar with this. So if I do slideshow, we're still seeing my screen, Ben? And sometimes you share and breaks everything when you go full screen. Are we still good? Okay.
Yeah, we're good.
So first, I'm just going to talk a little bit about like what metabolic model is and what it's not here. So what is a metabolic model? Just the simplest way of explaining is just you're going to get a list of reactions involved in the metabolic pathways for a given organism. It lists, you get some rules about the activity. So you get some rules that would tell you, something like this, where, okay, gene A performs function A, gene B performs this function. And then you get kind of a biomass reaction, which is just the basic building blocks for you to, for growth. Since we do try to do some simulations with these models, I can tell you, so a lot of people a lot of times they're just interested in 1 and 2. They just want the metabolic reconstruction. They just want to know what reactions, what pathways my organism is involved in, and they don't really care as far as going deep into running simulations, which is fine. So that's one of the biggest use cases. People are just interested in the reconstruction. So that's why we talk about this. What the model can do, you can try to predict some culture conditions. So some people may try to be designing a media for their organism to grow, may try to predict impact of perturbations, like doing some knockouts. So there's a lot of use cases, but that we can explore in a future webinar. But here, just kind of trying to go over what it's possible. So, and the key thing that you need to understand, like how this model is created. So your model is going to be as good as your genome annotation is. So at the key, what we do here is we look at your genome annotation that you submit. We look at the functions that are assigned to the genes, and then there are reactions that are mapped to these genes. So we have a template, what we call a model reconstruction template, where all of these mappings, right? That's kind of the sauce, and this is something you can go look at yourself and edit if you'd like in our publicly available data repositories. But this is basically what is happening. There's no magic. It's just we're mapping gene functional annotations to reactions. This is done. There's literature research. A lot of this is done in databases like KEGG and MetaPsych and models. So that the other big component is the biochemistry, right? This is the big one. And I have here, this is work that is led by our colleague Sam Seaver, which is the ModelSEED biochemistry database, where we basically look at KEGG and MetaPsych and BIG and MetaNex and Rhea and try to put it all together. And you might ask why, why not just use KEGG? Why not just use BIG and MetaPsych? So the problem that we have is that BIG is a good database as far because it's built specifically from modeling, but we want to get there's biochemistry data that is missing there. Our goal is to get more representation. And in KEGG and in MetaPsych, there's a lot of reactions that are not what we'd say genome scale metabolic modeling ready. So you have a lot of reactions that are abstract. They'll have generic acceptor and donor, described process that you just cannot model with these, and reactions that you cannot model with these models. So In a ModelSEED database, we end up getting all of that together. But when it comes to the modeling template that is used to build the models, a lot of that gets filtered out. It's modeling ready, biochemical reaction descriptions. But we try to integrate as much biochemistry as possible. And this in itself could be multiple webinars because this is a large problem. And then there's the ideal this cycle of annotation and validation that happens with these models. This is one of the use cases that we use in our own research and people do, where people build a model, they run experiments, and then they realize there's issues. And then when they go back and look, oh, the reason was because some annotation was wrong, or this annotation was unknown. This led to a gap in the model, and because of this gap in the model, we're not getting the prediction that we expect. So this is a common workflow and use case of these models. As I said, your model is going to be as good as your annotation is. And so that's why that's important. So here, I'm going to glance over this. So another key part here that happens in these models is flux balance analysis, which is a method that we use in reconstruction and analysis of the models, where we really try to explain here in very small detail. I'm not going to get into deep here. So basically, in order for me to give you a model that I can say that you can perform some optimizations and there's growth given in a source of nutrients, and giving them, which will be your media, say LB media, for example, and given the biomass formulation that we know that certain organisms need in order to grow. How is the model determining this? So this is like a basic cell representation, where you start with the nutrients, and then the nutrients will make their way through the cell, being converted from one reaction to the other. And we assume steady state, so we know this is wrong. So what that means is that you would have, everything goes from 1 to B and B to C, so there's no accumulation allowed. So you'd need other parameters. You need dynamic flux balance analysis to account for this. Then you're talking about enzyme constraints and kinetics that need to be accounted for. So in the type of modeling that we describe here, we assume a steady state. So basically something like this, where if you have 10 of a nutrient coming in, at the end everything is consumed, you would have 10 coming out. Or you could have something like this, where there's a byproduct, right? But at the end, all that came in still came out, right? There was no accumulation. All that came in, all came out of the system. So this is, I'm just gonna glance over this again. So what is the problem that you have in this? So this is quite simple, quite tractable in a small cell. Now think about this problem over, a network of a model can have upwards of thousands of reactions, thousands of compounds. How are we able, you can have cases where, particularly when you don't account for enzyme kinetics, where multiple pathways will have the same, could be chosen. How are they chosen? How do we know if these measurements are correct? What happens if there's gaps in the system? So I just told you about this, where unfortunately most annotations systems are incomplete, even for organisms, where we have plenty of knowledge. There's still gaps. Nowadays, I work a lot in the DOE space, and a lot of the DOE folks deal with large collection of MAGs. And so these metagenome extracted genomes have a lot of gaps, which make model building even more complex. So you end up more like a scenario like this, where there's a lot of pieces, where you just don't know everything, which means you end up with gaps, which basically means when you come to our cell, the moment if you don't know, this reaction, it just completely breaks everything. This is kind of just showing kind of the math, how the math is done in the steady state. But all that you need to know is that basically, if we have a gap, everything breaks down. We can't go from the beginning to the end, and your simulation doesn't work. So we need to figure out a way to fill these gaps. So when we go over to the narrative, you're going to hear about gap filling, and this is our process where we have our database, and then we try to, use algorithms to go in and try to fill these gaps, given what we know about the how those pathways should be completed. So that's just like a very small explanation there. And the last thing here on the slides before we jump to the narrative, I just want to give you a little primer talking about the ModelSEED2 reconstruction pipeline. Again, we're going to be focusing on reconstruction today, and what it does. Even though you don't see any of this when you use the app, all of this is going to happen all at once. But I would like you to understand the steps that are happening under the hood, which is the first step you need to do is you have to do an annotation with RAST. And you tell me why RAST. I already have my annotation that I bring into the system. We use RAST annotations because I'm not going to comment on the quality of an annotation system versus the other. I can tell you that RAST, from all of our analysis, they're very good at annotating genomes very consistently. A lot of annotation tools give you some very verbose descriptions. There's a lot of variation as they annotate the same functions. RAST is very good at giving you as a very good controlled vocabulary, and is very good at giving you consistent annotations across genomes. So that's why we like it for our template. Then there's some decision that happens. There's a machine learning algorithm that tries to detect what might be your organism. This is because we have a few templates. We have archaea and gram-negative and gram-positive. So this mostly will change the composition of your biomass equation, where, gram-negative versus gram-positive, there's a lot of cell wall compounds that need to be produced and accounted for. Archaea has a very different, will have a very different biomass reaction as well. And we are working on finalizing a cyanobacteria template that also has different requirements, where we need to account for that too. And then we build what we call the core model. What is a core model? Where we just try to get the energy biosynthesis working in a small model. And if that works, and if we get energy biosynthesis right, which is a very important step, and we move on to build the genome scale model. So I'm going to just jump out of here because I think it's important that we go to the narrative as our primer. I just wanted you to have a little bit of an introduction of how things work. And you can also feel free to email me and ask me questions. The other important thing I'll tell you would be this preprint, which hopefully with the paper will come out soon. But this is where basically every single methodology that you're going to hear today described and I already talked about is described in detail. So all these algorithms and steps and things, this is that image I just showed you, it's from the paper. So this is where I would go to find more information about the methodologies that I'm going to go over. So if you have the question doc, this will be a great time to look at, open this tutorial narrative here. You can go over here and do this right now. Let's go here. I already have mine open, but when you have your open, you're going to come here to this primer. And I'm going to start kind of walking you guys through this. And your narrative is probably going to look a little bit different because you can't. I have edit privileges. Yours, you probably won't be seeing this. If you're familiar with KBase, if I go to a tutorial where I don't have permissions, I would be able to actually kind of have the same vision that you have. So you probably are having kind of this view that I'm about to have, right? This is how you're going to see it. You can't do much, but if you want to kind of try to play with this yourself, you can hit this copy here and you create your own copy of that tutorial, and then immediately everything opens up. By that, I mean, you basically have the same view that I have, and you can kind of click in the apps. You can kind of reset the apps. You can add your own data. So it's a pretty good way. I wouldn't recommend you trying to follow along here. You could, but I would be more interested in you actually trying to do that later on, but you can do that. And that'll be a quick way for you to interacting with this. So we're going to start here just going over about our apps. So in this case, we're going to be working with E. coli. I know, kind of boring, but it just really helps us kind of showcase all that we want to showcase here today in this webinar. So the first step in this case, we loaded this genome in, and you can go about. There's plenty of documentation here. Then on our upload guides that you can go over to on how to upload data into KBase. So I'm not going to go into that. So we were able to load this genome, and you can see it here in your data panel. You see that there's two genomes, and one genome is the one that it was loaded, and then the other one you're going to see is this one here. You see it's the same genome, but as we call it RAST. So it's that first step where we bring the genome in and we annotate the genome with RAST. So it's very simple. You just load the RAST app, and then this is all default parameters where you were able just to annotate your genome with RAST. And then you can actually take a look at your genome that we just brought in. I click here, your genome will show up. And then you can kind of start seeing what that looks like. So if I search for something like pyruvate kinase, that's something that for sure we'll have, right? You have here, we know there's a gene that has this function, and then later on, we can search this gene, for example, and can see like, hopefully there was a reaction that was associated with this, with this organism, with this gene, right, in our model. But this is kind of, you can inspect your genome here, make sure that the annotations you expect are there. And once you have your genome, that's when we really jump into building our draft metabolic model. So first thing I would like to bring up here is, so what is a draft metabolic model? So why is it that we, oh, I guess, Ben, there's some people asking for access to the narrative or in the chat? Hey, let me know, yeah, if folks don't have access, but
Sure.
I'll continue here. So what is a draft metabolic model? So by building a draft metabolic model, it's just the first step where you're going from a genome to a list of the reactions that you are encoded to the genes and functions in those genomes, and you get those gene rules. You get those gene rules over there. So that we talked before. This is a model that it's not gap filled, so that's what we talked before. Meaning you won't be able to run simulations on a model, because even with something like E. coli, there'll always be a few gaps here. So you can read the descriptions here, very detailed descriptions for everything here that you have. But this is the app. So how you find the app, you would go here to MS2. You'd find the app. I would go here, click on the app, and if this is what pops up. We can talk a little bit what you can do here. So you can add your genome here, or a genome set if you have that. You can add your medias to for gap filling. We'll talk about this when it is relevant. If you don't add a media, it defaults to what we call doxo media, which is a rich media, which you can kind of look at our documentation, what it does entail. But more importantly, in this case, remember, we're only trying to build a draft model, so we uncheck this box. We're not going to gap fill it. We just want to have the representation without any algorithm adding more reactions to our model. We just want the mapping of genes, functions to reactions in our E. coli genome. You have a few advanced parameters here that you can look. So I talked about the templates. If you saw, in this case, you could see what templates are available. In this case, we do the automated selection. It's what I meant when there's a machine learning algorithm that tries to look at your genome and tries to figure out if it's a gram negative or gram positive or archaea. but you can just set it yourself. If you know already what it is and you don't want to have to deal, with an algorithm possibly making a mistake, which it can, you can set it yourself. There is a situation where we see that, for example, if you try to build an organism, a model for a cyanobacteria, you'll get a warning saying that it's not possible, that you don't have a template for cyanobacteria. You can bypass that here by selecting your own custom template. You just won't get a very meaningful model. I also know that classifier is a little sensitive right now. So there is instances where they misclassifying your genome as a cyanobacteria. So that would be another reason for you to come here if you're getting that warning and to select a different template yourself. So pretty simple here. So we're gonna go and hide principal here. So I'm gonna delete this because we have the magic of already having this app here already filled with our E. coli genome. We are calling it our draft model. And then once you hit run, you'd get an output and yeah, you get your model. So you get your model, you immediately get some stats here about your model. But the more important here is we can go down and you can talk about here a little bit about how you go about reading this output. I'm going to open the model that I get over here, but you have the primer here down, all the information in detail about it. So when I look at the model, as I said, the main thing that you get when you build a model is a particular draft model is this. You get the mapping of reactions to functions to reactions. So you can see here, you have this reaction was mapped to this specific gene. I'm actually curious, like what we talked earlier, like let's see about this gene here, or pyruvate kinase gene. I can go to genes here and search for it. And you can see there's multiple reactions that are mapped to it. You can search for the gene here, and you can see these reactions here. So that's working as expected. But yeah, so you get an overview, just some stats about your model. You get the full list of reactions about your model. You get the full list of compounds that are present. You get the list of genes and associated reactions with those genes. You can click on a reaction and open up, and you get some more details about that reaction. You get the list of compartments here. This is very basic here because we only have basically the exchange between the cytosol and the extracellular. There are other resources that account for the periplasm compartment, which we don't do, mostly because in a lot of cases, it just adds complexity. It would be more accurate to have the periplasm concept, but the majority of what happens in the periplasm, what you have is just a bunch of duplicated transport reactions. So right now we have a reaction that goes from the cytosol to the extracellular, and if you represent the periplasm, you'd have one extra reaction to represent the movement between from the cytosol to the periplasm, and then from the periplasm to the extracellular. So there are some processes that only happen in the periplasm that would get better represented by us having a periplasm compartment, but it's not a deal breaker. It's for the level of abstraction that we're working on. It's not. You're not getting worse or better results in your reconstruction because you're not representing that compartment. So you get those, and you like your biomass, right? We talk about all these compounds that we have here that are necessary for growth, that they need to be produced for the model. You see that gap filling is empty because we didn't do any gap filling. And then we look at pathways here. We have this is kind of a legacy KEGG map visualization, but you can kind of see what ended up in the model. The problem with KEGG maps is that they're static and they may not represent all organisms in the same way, but this at least gives you an overview. This is also a little bit outdated because there's just some issues with KEGG licensing and how we can distribute these maps and update them. So these may not be the latest updated maps. We have to revisit this internally to see how much value it adds or not, and possibly deal with licensing issues. So that's why you may not see the latest maps here, but I don't want to get into details in there. but it's here if you want to look, kind of have an overview of what ended up in your model. So this is kind of an overview of your draft model, of your draft model here. So, and you have your detailed descriptions below. Then we talk about gap filling the model. So this is the part where, okay, can we make the model grow in a minimal media, in this case in glucose minimal media? And you can see here, if you select by medias, that the glucose minimal media is available. You can even look at it, like it's here. You can see all the compounds that are available, right? It just basically mostly just trace metals and glucose. In this case, we have oxygen too. And you can also see here something that is important for you to account for. You can see the minimum and max uptake. Here, these are the bounds of the media. You can see that glucose is limited and also oxygen. So this is something that you kind of have to play yourself with when you're doing simulations. You're trying to understand for your organism and system what quantities of a carbon source or what quantities of oxygen might be limiting. So these do not correspond to an exact value or concentration. Again, concentration doesn't even take it into account here because we operate at a steady state level where there's no accumulation, right? So basically here is just a way for you to like imagine if you're trying to, as you play with these bounds and edit your media, if when you're doing your simulations, you're looking at the value of transport of glucose into the cell and you're trying to see, oh, is the constraint that I set already limiting, right? Is or because if you don't limit the consumption of glucose, which is usually limited in your systems, the model is just going to make as much use of it as it can, and then your flux distribution is not going to be realistic because the model is going to be allowed to eat a large amount of glucose that they wouldn't be in reality in your experiment. So these are things that you have to play with these constraints. Same thing with oxygen. So this is something that we can also dig into a future webinar. But here's something for you to be aware, how the media constraints impact your modeling simulation. So where do we got this media from? If you go here and you go to public and you go to medias, we have a decent amount of medias that are available. This one was this carbon D-glucose, but there's a bunch of minimal medias that you'll find for all sorts of different compounds. They're helpful a lot of times just for you can start one of these medias and then edit. There'll be an edit media app that exists here. And if you need to start, you can start with this carbon D-glucose, and then then actually I want to just remove the oxygen from this media because I want to do an aerobic simulation. So you could start with this media and edit it and remove oxygen completely, or you could just set the constraints to 0, and then oxygen will still be in your media, but it wouldn't be used. So there's different ways of doing this. So what is the main difference now? When we go to our metabolic modeling app, you still select your genome, but now you're selecting your media here for gap filling. And the gap filling flag is turned on here now, or our checkbox. So after you build your model, you're going to get a nice report here about what happened. As you can see now, before we actually have gap filling, it's saying that we have 4 gap filled reactions, and then you also get your growth simulation here, your value, which is calculated in this moles program for like of dry weight per hour unit. And I'm actually, but what you get too is you get some cool reports that I kind of want to dig into. You can get this full view, which is basically just like a larger page that opens with, but it's mostly that information that you already have. But I want to actually dig into this full reconstruction report, which opens a separate page, but you also have a little bit of an explanation. But under here, like I'm not going to go under, I'm not going to review it because I'm just going to detail myself, but this is pretty basically all I'm going to describe now is nicely explained and described on here. So, but first, so what is this report tells you? So the reason I created this report is because this reflects a lot of the questions that people had about some of the modeling being a little bit of a black box, and they really not understanding what happens, because you just hit a button, and then there's a model that comes out at the end, and you don't really know what happened. So first here you get something. The first information that you're getting here is you're getting this thing that it says core gap filling media, no gap filling needed at the core. So this is good news. So basically, we did a lot of curation, and you can read that in the paper, to curate the central carbon and energy metabolism in our core models. So ideally, this means that in order for your model just to produce ATP, nothing else, just to produce ATP, and ideally produce a realistic ATP amount that it should be, as is reported in the literature, no gap filling was necessary. So this should be the majority of the cases. If your organism requires core gap filling, it means it just might be some it's like a lifestyle that we might not have properly represented in our templates that we need to curate and add. So, for example, recently we had to work on getting sulfate reduction bacteria metabolism encoded. So that's one of the things that we have to add. We're working with a group to do iron oxidation. So that's stuff that we have to add that is unique. So but usually it's a good sign that didn't happen. If you need gap filling at the core, it usually means your organism is either incomplete, or it's an organism with biochemistry that we don't have properly represented in our templates, which means you may shoot us an email, and with time and research, we might add it, because this is the other aspect. There's the aspect of developing these apps, but there's also the aspect of doing the underneath science of curating. There's a lot of manually curation that goes into these templates, and assigning reactions and functions. So, and it just takes time to do as we develop the apps themselves as well. So you get some statistics about the number of reactions that you have in your model, and compartments and gap fills. So, and then you get this gap filling analysis. So this is something that is important for you to understand, because you need to. So why is it that a certain reaction? In this case, we had 4 reactions that were added by gap filling. Meaning, if these reactions were not added to the model would not grow on glucose minimal media. So what is this? We're trying to tell. So the first one that I'm seeing here is, you see an exchange for this 5-dimethyl D-ribose, which is out. So it's to export this out of the cell. So what is happening here? So what is happening here is that it seems, in order to, and you see here the sensitivity ID and name, basically spermidine is one of the biomass compounds that this organism needs to produce in order to grow. So it basically is telling you, in order for spermidine to be produced, this compound needs to be excreted out of the cell. And you see here that another reaction that was added is this 5-dimethyl transport. So first we add the transport out, and then there's an added exchange reaction that is added. And so when I see this, I was actually curious, and so I actually looked at the E. coli models, and in the published models, and one of the previous versions of the earlier versions of the E. coli models, actually, they also added this exchange added to get rid of this compound, because the reactions that were necessary to kind of recycle this compound in a model did not exist. And if you remember, in steady state, as we're trying to build these models and we do the flux simulations, nothing gets accumulated. So basically what was happening is the template didn't have the proper reactions in order to excrete this compound, to recycle this compound. So the solution that the gap filling found was, okay, we're just going to get rid of it. I have curated the template in order to make this, because some of the functions are present, but some of these functions are not annotated properly in all genomes. For example, if you try with other organisms that recycle this compound in a different way, you won't see this. I know that if I try with Bicetullus, for example, when I do this, you won't see this gap filling reaction, because they have a different way of handling the recycling of this compound, and that pathway is better annotated, so it gets into the model. So, but these results also help us to go and to fix the template. And in some cases, even if I fix the template, the algorithm, that's the other thing, the algorithm might need to be tuned because the algorithm still prefers to do this excretion over, the 4 or 5 extra steps to recycle it. So you may need to force those constraints. So this is all part of working with these models, and more importantly, understanding what the gap filling does and understanding that this is not critical, right? This is not something that is going to break your model simulations if you understand why these reactions were added. And then there's a couple other reactions here that also get gap filled in, that also have to do with either poor annotation with this genome or representation in the template that need to be adjusted, or a mix of both. But more important, you kind of know why these reactions were added, what biomass compounds they trying to add in order to accomplish this, and so we have an idea. You just don't blindly understand, okay, gap filling, try to do this. And then you get this core ATP analysis here. So basically, when we build a core model, we test a bunch of conditions that you see here in order to see if ATP can be produced. And, of course, ATP can be produced here with glucose in the anaerobic media, which is kind of what we were testing in our genome scale media that we provided. But you can kind of see here, see basically in all these conditions is able to produce ATP. But then you also start seeing this where, okay, in succinate, even though this was not added to the model, but if you were trying to build a model, grow the model in succinate oxygen O2 media, it's only possible if this one gap fill reaction is added. When there's only one gap fill reaction that is necessary to do anything, I started having doubts myself. Is this real? Does this organism is actually capable of growing in succinate and O2 media, minimal media? Or was it just a problem with annotation? So this kind of helps you having that idea. But once you start getting things like this, where, in order to do sulfate reduction, you need to add 11 reactions, I think it's pretty clear that he's telling you that, this organism can't do sulfate reaction. And you can see here some of these, methylamines and other things. So this is kind of gives you an idea of the metabolic capabilities and the lifestyle of the organisms you're trying to model. Particularly, this might be more helpful if you're dealing with MAGs or large sets of organisms that you don't really know what they are and what they do. The rule of thumb here is if you need a lot of gap filling to get energy biosynthesis working, either your genome is in really bad shape or your organism just doesn't do it. So that's kind of the takeaway that I would like you to get from that. So this is probably one of the most important things for you to understand, and was one of the main things that I put a lot of work, and is probably one of the key things in this ModelSEED2 release that we did. If you read the preprint and the paper when it comes out, we really went in and wanted to make sure you get energy biosynthesis right, because it's very common that you get energy generation loops. This was very common in our previous version, and where the FDA can exploit some unlimited ATP production. And if that happens, then all of your entire flux distribution will be meaningless, because your model will be doing things that your organism will never do, because your organism will never have unlimited ATP. So that's why we really try to nail down this core reconstruction before we move on to genome scale. I see some questions here already, but yeah, you can add your own media. Yeah, you can add as many. You can create a media from scratch. I'm not going to go into detail in that here now, but yes, you'll find documentation of how to either start from one of our medias or fully create your own media from scratch. You can even create it in a CSV file in your computer and upload it. Yeah, you can use whatever media. If you don't see a media available there, it doesn't really matter. You can create whatever media you'd like. That's not a limitation. So hopefully that became clear here on this analysis. And then the other aspect here is that if you build a model, is you do this, you run this basic flux balance analysis, which I kind of went over. So you can run as many simulations you'd like. The process of, in this case, as you can see, we're selecting this gap model that we gap filled, right? And then you were selecting this carbon D-glucose media that we went to get. By default, we're trying to optimize biomass. So this is a key tenet of flux balance analysis that relies on the assumption that all organisms have evolved in order to grow and produce more of themselves. So that's how we are operating here. But you can try to select, imagine, oh, I actually want to optimize for the production of ethanol. So you would select the excretion of ethanol could be your objective here, your reaction to maximize, and then you would have completely different results, right? So you can think of other objective functions that we would have. By default, we try to maximize biomass. So when you do this, you get, you get your flux distribution, which, it's, you get your results. So this is something that you kind of really need to try to understand. If I sort through flux here, you can see that there are reactions that will have flux. If you see a reaction that doesn't have any flux, it basically will tell you that is a reaction that is inactive. So a lot of reactions in the network, will never have any flux. Might never have any flux. So you can see, you can look through that here. Why is it that you might have different reactions? You can see the exchange fluxes. So this is the fluxes this is important for you to see what is actually, just because something might be in the media, might not be fully used. You can kind of see how much, we talked about earlier about stuff like glucose, for example, right? And if I look at my glucose exchange here, you can see that, I'm setting it up to 5 as the maximum and is using all of it, right? It's telling me here flux through this is using all of it. While you can see here, for example, for others, even though they are constrained to a value of 100, they're not really fully used. But here, glucose is. So you can play with this. You can play with this. If I set it to 10, would it still fully use it? Like, what is the rate limit that I want to use? Are my simulations getting more realistic as I play with these values? So it's all part of understanding how flux balance analysis works. Like, we could do a full webinar or multiple webinars just on flux balance analysis itself. I don't want to get into many details here because that's mostly about you understanding that. And then here, understanding just reconstruction at this point. And then here you get a little bit of that map, but here you get painted. You still have your presence and absence. In this case, you can see, I guess there's presence. You can see in these genes are present in your model, but it's basically telling you there's no flux through those genes. And here, your red and green, you have basically a consumption or production of a certain, enzyme being used by the model. So this is what you get from your flux balance analysis. And we also are trying to use Escher pathway viewer here, just to give you a little bit more granularity. So these maps would actually show you the reactions that are in the model instead of a static CAG map like you see here. This is still a work in progress where you can try to change here and if you look at the app here, you'd see that we have a catalog of maps to look through. But this is, these are arguably not the prettiest maps because these were manually building these maps. But these will be, help you get a better way. The thickness of the arrows. The goal here is to show you more or less flux through that, right? We also have options to overlay gene expression data with your simulation. Again, that's another advanced use case that if you have expression data, we can use it to constrain the flux simulation. But that, that could be another part of our series on metabolic modeling workshops. But I just want you to know that that's here, and you can look at these maps, and sometimes they might be more helpful than the CAG maps as far as providing you information of flux through a certain pathway. So I want to leave some room at the end now. I don't want to get into many more details. I don't know if we have what is our structure for questions, Ben, if we're just going to go through the live document and answer at the end, or if we have some room from some questions now, how you want me to Yeah.
There was a few questions. I answered a lot of them, but there's a few without answers. And one Anuradha was asking about with setting the uptake constraints. Do we are those set as plus minus 100 as a standard, or are those sort of derived from experimental studies? What's the kind of basis for setting those?
Yeah, yeah, it's not even a standard, it's just a value. Again, like some people set them as minus 1,000 to 1,000, because basically what that means is basically means that you are unconstrained. Those resources are not constrained, right? They're able, but when you set a value like that, you basically able to let the model to uptake those as it pleases. And in a lot of cases, it's not an issue. As you saw in the FBA simulation, the model will intelligently like make use of the resources, but in something like your carbon source, we'll for sure try to exploit it, right? Remember what flex balance analysis is trying to do when we're maximizing biomass is trying to make as more biomass as possible. So if the glucose is key for biomass consumption and it's unconstrained, it's going to use as much as you give it. I guess if you put 100, I'm pretty much sure it will use 100. If you put 1,000, it might not use 1,000, right? That's kind of a way of you kind of seeing how this works. But if you look at metabolic modeling workflows, these are very common. Just like some basically, just by having those volumes for those numbers for excretion and uptake, you're basically saying it's unconstrained. It's nothing derived from any experimentation. It's basically, it's part of the trial and error of working with these models. It's trying to understand, what constraints are limiting for certain nutrients and how you can, because ideally you're trying to attune to your experimental data as much as possible with your simulations.
Great. Another one as just kind of your perspective on using KBase for draft model production versus like Carve Me, Raven Toolbox, Cobra Pi, those kind of things.
I mean, so I can tell you, so you can see actually in the preprint, we do a comparison with CarveMe. Um, there's advantages and disadvantages to both. Like I could go into details there, but we actually really went into detail into the paper. CarveMe has some advantages. It's just a different approach that leads to like, so we see that for example, one of the main things is we get more diverse models across phylogeny with our template approach versus the universal model approach that you see in CogMe. That's one thing. We see that we get better results for the energy biosynthesis that I went over, where our values seem to be more realistic versus CogMe. But currently, like you get models without any gap filling, right? Because they're using this, which saves you the trouble of trying to understand the gap filling and if the gap filling results are correct, right? Because the gap filling, I always say the gap filling, it's more like a zip code than an address. It doesn't have the full context of an organism. So we might the organism might cheat, right? In order to complete the step, might add one reaction, while your organism in reality probably has an alternative pathways that is 3 or 4 steps. But if they are more incomplete, the algorithm tries to add the least amount of reactions as possible. So with CardMe, usually are able to get models without less gap filling, because they're going over a universal model built out of these big manually curated models. But I can tell you from experience, and I know if you guys have experience here, a lot of these, manually curated genome scale models are not that great unless you're talking about the E. coli model, the Saccharomyces models, the true model organism. Because what happens with a lot of people is they'll start with a tool like CardMe, they start with a tool like ModelSEED, and they build a draft model. And then in their research project, they're interested usually in studying a few pathways, right? That is their pathways of interest. And in those pathways, they probably really curated those pathways in that published model, which is great. But they didn't went through the whole full genome scale model in making sure that every pathway and flux distribution through all those pathways was realistic for the whole model. No one does that unless you're talking about, the true like model organisms. So just a warning, like, because we see a lot of because we look at these publisher models as a reference, and we see a lot of issues in publisher models just because there's parts of the model that they didn't care, right? It was not part of the problem they're trying to answer, but they still publish as a genome scale metabolic model, but where a large portion was mostly built by one of these tools. And the level of validation that they do to truly call it a fully curated genome scale metabolic model varies. So yeah, like for sure, when you talk about tools like CobraPi, we for sure limited still in our what you can do after the model. So, after you build your model, we see, it's clear that people, you can download your model in, SPML format and go do your analysis where you think it's best. So that's up to us to implement more analysis tools than just basic flux valent analysis. So that's definitely a workflow that we see, that people download their draft model and then go and take it to other tool to do gap filling if they would like, or to do simulations. But yeah, there's advantages and disadvantages to both when you're doing metabolic model reconstruction. And the key lies into, I know that's people that build with both. They build with CardMeet, they build with ModelSEED, and then they try to merge it in. I know that people try to do that when they're trying to get the best model possible. It really depends on what you're trying to accomplish with your metabolic reconstruction study and analysis.
Mm-hmm. Another question is there a high throughput way to run metabolic model reconstruction for multiple genomes? Yeah, so the batch function.
Yeah, there is a batch you can see in the app for if you create a genome set. There is an option. There is also, we have a way for you to kind of run this programmatically that it's not fully exposed because there's some issues and requires some more advanced knowledge. If you are someone that wants to do that, just reach out. If you contact the KBase help desk, they will immediately point you out to that, right? You go to support and go to the help board, you will immediately get help. You will immediately get help about how to get that set up. So it's possible. Actually, you can do a lot. You can run, thousands of FBA simulations too. I've done that, but that's just not fully exposed and tutorialized at this point. But if you are someone that is interested, just reach out to us and we'll set you up.
Mm-hmm.
But just the basic here, yeah, you can build a model here. Just you saw in the model building app, there was just a basic here, no genome or genome set. You can at least do that pretty easily.
Mm-hmm. Is there a way to manually alter reactions in the draft model instead of running the algorithmic approach?
Oh, so there is a edit metabolic model app that exists. To be fair, it's old. It's a little clunky. You might have more issues then you might just, it's not ideal for us because we lose provenance, but people just kind of download the draft and manually edit the model locally. You can bring it back into KBase. You can download and re-import. That clearly shows we're lacking in this department. But editing a model, it's really hard. Like, the problem is that this app, there's just so many boxes, so many things to account. One of the big problems we had is that people make a lot of mistakes and then a model doesn't work because there's so many things that you can input wrong that we have to validate. And then it's just a hard problem, just to provide a satisfying user experience. Then at the end, you're probably better off just doing it manually. And then when you try to bring the model into KBase and upload it, there is a validator that if there's something wrong with your model, the import would fail. But it's just a hard problem. It's here, it works. It's just not the greatest experience. That's not why we heavily, just feel definitely an opportunity here for someone, for a project for that to be done here. But currently not the greatest experience. And we see that folks usually like to just kind of, download locally and re-upload.
Yeah, absolutely. If you have any desire to help us work on KBase apps, please reach out to us and we can help you do that with us. Yeah, another one is just about nutrient uptake constraints, like looking to see if there's a database available to see, some experimental values used for that. I assume you would just have to kind of look in different publications for.
I just look at publications. Yeah, there's a body of literature, and you can kind of see what people run these simulations and stuff like that. Yeah, that's just something. Yeah, There's no way around it.
Mm-hmm. Someone asks, are these models able to predict with some confidence more specialized chemistries like compounds from biosynthetic gene clusters, or more useful for predicting precursor molecules for those pathways?
Some of those things might be possible depending on, using kind of add-on tools to improve simulations. So yeah, it's possible, but not necessarily with like all that it's in here, right? It just requires a little bit more. I can't think on the top of my head, but if you, I mean, I'm not going to say Google it, right? But there's some tools out there that allow you to do that more with ease. I just can't think of the top of my head. But if you have a specific workflow you want to do, feel free to message the help board, and we can then do a more thorough analysis and like, yes, what you want to do is possible. It's possible within KBase, but it's not. I have no problems of sending people to do workflows outside of KBase when we're lacking either.
And there is a beta version of anti-smash that you can use at least identifying those based off of the genome annotations. But yeah, it's not directly related to this. Yeah, one last question is, what are the kind of downstream analyses that you can do with these models in FBA? Models like, yeah, what do you see as the next step for people?
I mean, downstream analysis, like a lot of people, what they like to do is I mean, they try. So there's a range of first just the qualitative versus quantitative analysis here, right? Where people are trying to really like dive in and see if they get the exact, growth values that they would get in their simulations, right? Or something close to it. So those are more difficult to tune, but there's a lot of people that are just like interested in the more just quantitative, like qualitative versus quantitative, where like I just want to know if something grows or it doesn't grow. Um, that's are like I just want to see if this organism is able to perform, this process. It has this pathway and is able to perform this process. So that's much easier to accomplish without having to really, dig in and into tune your FBA simulations. But like it's very common. It's very common that, for example, people are trying to design media, right? They may have an algorithm, an organism that they're having trouble culturing, or they want to culture, but the media is more expensive. Is that some components are expensive, or some components are not ideal for what they're trying to do? So they like to try to design some alternative media that would still allow them to grow at a decent yield that would like serve their research purposes better. So those are some of the analysis that people like to do. People like to optimize production. So strain optimization, it's like big use of these, right? Thinking about all sorts of metabolic engineering opportunities where you're trying to do gene deletions, single gene deletions, multiple knockout simulations, and trying to do pathway and strain optimizations. Those are some of the classic uses that people have. And then I didn't touch into this, but like a lot of days we also do a lot of community modeling, right? Where you can actually merge multiple models together and simulate model interactions. So that's in another level that will be an even more advanced webinar.
hmm has a very specific question about a
I
certain
don't know.
yeah.
I've seen a
Yeah.
certain media. I see that. I have no idea. I'm not a
I don't
mycobacterium
know.
expert. I have no idea what that medium is, why, how it was added there, unfortunately.
And one
So,
last question is
yep.
about doing this for mags. I guess kind of generally, what would you kind of say for, using
Yes,
mags?
it's possible. So again, so this is when you're looking at mags, and we actually have there's multiple. I mean, there's a few papers where we did this. So I can point you to there's the Grow paper on Nature where we were collaborators where we did mags. There's the There's the Nature Biotech paper that we did too with JGI. So we've done this, but but you have to. There's a lot of work that needs to be done with enriching these mags. A lot of these mags are incomplete, so there's a lot of work done in the community for pangenome enrichment of these mags. So because at the end of the day is garbage in, garbage out. As I said, your quality of your model is going to vary tremendously depending on the quality of your genome, of the genome that you bring in. So there's a lot that can be done. You can look at those papers. If you can't find the grow like it's like river water analysis paper, and so there's a few papers that I could point people to, but where we did those large scale mag analysis. But it's a difficult problem, and it's honestly building the models ends up being the easier part. The difficult part is to get good mags.
Mm-hmm.
Another
Yeah.
question here I'm seeing. So when you talk about is always preferable to use defined media always, because there's no such thing like it's a common question that we get in our pipeline work here is people are getting help with their media. If you add, for example, if you have a media that is bovine serum is not something that you can put in your media, right? So what people have done, they try to approximate. If you look at our, LB media description, it's in a sense the defined media because, we try to figure out what is it that bovine serum is providing to the media, right? Maybe some vitamins is something, right? So you can't so people also try to put like complex. So if you have like NaCl, right? You can put NaCl in your media. You put Na, and you can't put like salts in crystallized form. They need to be in that aqueous version. So you need to think about when you put something into the media, how is that the pathways these reactions are going to uptake these compounds. You can't just put something that just unrealistic, right? That the biochemistry doesn't respond. So if you search through the help board, I think this is a common question, and search for media. I have plenty of tickets where I actually go through people trying to help them answer this exact question because they're trying to figure out how to create their media. How to because they say, oh, I tried to represent this media and it's not working. So this is a very common workflow that we have, is that, oh, it's not working, and then I look, oh yeah, you have a bunch of non-defined compounds in your media, of course it's not going to work.
Great, thank you. Let's see if there's any other ones. A request for a FBA webinar, so we could do that maybe early next year. Let's see. Can KBase connect two chemical compounds and show potential biochemistry synthesis pathway via de novo synthesis method?
That's I'm not sure about that. It's like yeah, I mean, not in KBase. You can do those type of analysis, but not in KBase. No. So when you talk about accounting for pH, there is not a way to account for pH. So all of our reactions are set to the neutral pH. So the only way to do that is, and it's something that we'll love to do eventually, is that you basically have to retune the entire chemistry to calculate. We would have to say calculate the all these pKa values to a range of pHs, and then we would have to say in your simulation, hey, actually, I want to simulate this at a pH of 6, and then in a sense, you would be using a completely different database than what we're using right now, because this can impact reaction directionalities, and there's all the implications of pH. So there's no way to account for that right now in the current state of the biochemistry that is only tuned for neutral pH. So that's basically how you do that. I mean, we have thought about this. It's kind of a dream. I would like to have that for our own research, but it's a very difficult problem.
Great. Yeah, I think that, let's see, maybe anything else? Yeah, all right, great. Thanks for asking all the questions. I'm just responding to one in the chat now, but yeah, we will post this webinar up on our YouTube. And of course, we'll keep the questions and answers document open for you all to revisit in time. We posted some of the links to the papers that Jose was referring to in there if you want to do more exploration. But otherwise, thanks so much, Jose. That was a great webinar and great introduction to metabolic modeling and flux balance analysis. So we'll plan out some more of these next year. And if you all have specific things you would like us to cover, you can reach out to us at engage@kbase.us or reach out to us via the help board. And yeah, hopefully we will see you all successfully building your own genome scale metabolic models and publishing them yeah, in the literature. So feel free to reach out to us if you have any more questions or inquiries. And other than that, I think we can stop the recording.
Thank you, everyone. Thank you for taking the time to join us today.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.